Welcome to Urinalysis, Part 1. This is a capstone review for urinalysis and body fluids. My name is khieb. I am the MLT Campus Coordinator for the Bismarck Campus. This is a 4-part series. Part 1 is only going to be approximately 25 minutes long. And with that, we'll get started. Here are the topics we're going to review today. Review the importance of urine, composition of urine, review renal anatomy and physiology, discuss how urine is formed, summarize endocrine functions associated with the kidneys, and review renal function tests. Just a little bit of perspective here. Just so you know that your urinalysis and body fluid section of your Board of Certification exam is only going to be about 9%. I have all of the percentages listed here for you. This certainly is an important topic, but it definitely doesn't comprise very much of the exam. So take that with a grain of salt when you're studying for your boards, and even though this might be your very, very favorite topic, you want to spend more time on the other sections, definitely. I wish I could tell you what questions are going to be on your boards, but we do not know that information. This is simply an outline of what we're going to cover in the next 4 review sessions. Go over urinalysis in general, complete urinalysis, including physical, chemical, microscopic tests, physiology of the kidneys, and finally review body fluids. So why is urine important in the first place? Why do we even care? This is one of the questions that I ask constantly in my classroom. Why do we care? Why do you need to know this? Well, you probably recall from your course and your practicum experience, urine is a way for us to eliminate waste products from our bodies that build up. And changes in urine can be and are directly related to disease states. And urinalysis is used to diagnose and monitor disease. It's also a great thing to use for drug screening, and is also used to diagnose inborn errors of metabolism. Urine is mostly comprised of water. There are other dissolved organic and inorganic compounds. The composition of urine is affected by diet, how active a person is, what kind of metabolism they have, and if they possess any disease processes. The composition of urine is also directly related to the amount and the type of waste material that needs to be excreted. So that also has to do with the type of metabolism that someone has. I simply have listed here the organic substances found in urine. The top 3, urea, creatinine, and uric acid, are found in larger amounts, and the others, glucose, protein, hormones, vitamins, and metabolized medications, are found in much lesser amounts in the urine. Inorganic substances found in the urine include this list: chloride, sodium, potassium, found in larger amounts, sulfate, phosphate, ammonium, calcium, magnesium, found in smaller amounts. Non-dissolved substances, normally we see these when we do a microscopic: bacteria, crystals, casts, mucus, various other types of cells. So let's talk a little bit about renal anatomy. As you might recall, the kidneys are 2 bean-shaped organs located under the diaphragm on either side of the aorta in the posterior upper abdominal region. You have an outer and an inner area of the kidney. The outer portion is called the cortex, and it is comprised of renal corpuscles and the proximal and distal convoluted tubules. The middle or inner part of the kidney contains the luce of Henle and the collecting ducts. In addition to the kidneys, we also have two ureters that are muscular tubes that connect the pelvis of the kidney to the bladder. The bladder is where urine is stored until it gets excreted through the urethra. The abdominal aorta supplies blood to the renal artery, which in turn provides blood to the kidney, and the renal vein returns blood to the inferior vena cava. This is just a schematic for you to take a look and review the anatomy of the renal system. This diagram shows you the same structures, except it shows you a little bit better area of the nephrons and the parts of the kidney like the medulla and the cortex, the inner and the outer areas of the kidney. So perhaps the most important part of the kidney is the nephron, and it certainly is the functional unit of the kidney and is key in urine formation. So the nephron consists of renal corpuscles, the glomerulus, the Bowman's capsule, and a very complex tubular system. The glomerulus is a tuft of capillaries that lie in a tubular depression called the Bowman's capsule. You have an afferent arteriole and an efferent arteriole that supplies the blood to and from the glomerulus. And then you have peritubular capillaries which stem from the efferent arteriole, which also have to do with the reabsorption process that takes place in the glomerulus. The main function of the glomerulus is to filter blood. The proximal convoluted tubule is located in the cortex. The loop of Henle begins in the cortex with the descending limb of the loop extending into the medulla where the bend of the loop is formed. Then that becomes the ascending limb, which ends in the cortex. The distal convoluted tubule is located in the cortex, and this is what forms multiple nephrons, and they direct the flow of the urine into the collecting duct. The collecting duct joins with other collecting ducts, and that joins to the renal pelvis, which ultimately leads into the bladder. So there's three major processes that take place in the kidney: glomerular filtration, tubular reabsorption, and tubular secretion. The glomerulus is a semi-permeable membrane, which means large molecules stay out, they're unable to get through, and small molecules are allowed in. Large molecules like protein and cells, smaller molecules such as glucose, urea, sodium, etc. So what this does, because it's semi-permeable, means that an ultrafiltrate of the plasma forms. From the glomerulus, that ultrafiltrate flows into the proximal convoluted tubule. About 115 to 125 milliliters of this filtrate is formed per minute by the glomeruli, and all but 1 milliliter of this ends up being reabsorbed by the renal tubules. By the way, if you didn't remember this already, we talk about the nephron as being the functional unit. There are literally millions of nephrons found in each kidney, so they're all reabsorbing and acting as a semi-permeable membrane for the filtration of serum or plasma, and all with the function of concentrating urine and creating urine. Some definitions you should know: what reabsorption and secretion are. Reabsorption is that process by which filtered water ions and molecules leave the tubes of the tubule system for return to the blood. So if your body is in need of something, sodium or water or whatever, it can be returned to the blood. Secretion is where a substance from the blood is transported across the wall of the tubule into the filtrate. The proximal convoluted tubule, this is where most of the reabsorption and secretion occurs. There is a renal threshold here. What that means is there is a limitation as to how much solute can be reabsorbed. In the proximal convoluted tubule, water, sodium, chloride, potassium, urea, glucose, and amino acids all get absorbed or reabsorbed. Hydrogen ions and medications will be secreted here. The loop of Henle has a descending limb and an ascending limb. In the descending limb, water gets reabsorbed there. In the ascending limb, sodium and chloride get reabsorbed. From the loop of Henle, the filtrate goes on to the distal convoluted tubule. Of note, 85% of all reabsorption takes place before that filtrate ever gets to the distal convoluted tubule. So a lot of activity going on in the glomerulus, in the proximal tubule, and the loop of Henle before it ever gets to that distal convoluted tubule. Here, the sodium is reabsorbed here, and hydrogen and potassium are secreted. The reabsorption of water is controlled by antidiuretic hormone in the distal convoluted tubule, and the reabsorption of water and sodium is controlled by aldosterone, and that secretion of the potassium and hydrogen ions are also controlled by aldosterone. And we'll talk about those hormones a little bit more later. The collecting duct is the final site for water reabsorption. So if your body still needs to hang on to some of that water, this is the last chance for that to happen. This is under control of antidiuretic hormone. Aldosterone is also present, controlling the reabsorption of sodium and chloride. In the collecting duct, osmosis also plays a role in water reabsorption. I'm going to talk a little bit now about some hormone activities that are associated with the kidney system. The renin-angiotensin-aldosterone axis. Renin is secreted by the juxtaglomerular apparatus of the kidneys and catalyzes the conversion of angiotensinogen to angiotensin 1. Angiotensin 1 is not active at this time. From there, angiotensin 1 stimulates the production of angiotensin 2, which is the active form. Angiotensin 2 regulates renal blood flow by constricting the renal arterioles and also secreting aldosterone. That aldosterone, in turn, facilitates the retention of sodium. Aldosterone is made in the cortex of the adrenal glands. Remember, the adrenal glands are right on top of the kidney. You can see the diagram there. The aldosterone acts on the kidney by promoting the reabsorption of sodium and secreting potassium. Note that water also gets reabsorbed with that sodium under the control of aldosterone. Antidiuretic hormone is secreted by the posterior pituitary gland. It promotes water reabsorption, and ADH primarily affects the reabsorption of water from the distal convoluted tubule and the collecting duct. Parathyroid hormone is made in the parathyroid glands. This hormone promotes calcium reabsorption and the excretion of phosphate. So there is an indirect relationship or an inverse relationship between calcium and phosphate. And last, erythropoietin is a hormone that is produced by the peritubular fibroblasts in the kidneys, and what this does is stimulate red blood cell production in response to hypoxia. So when your body does not have enough oxygen, the kidneys are stimulated to produce erythropoietin, which acts directly on the bone marrow to tell the body to make more red blood cells to try to correct the oxygen imbalance. So the next few slides, we're just going to talk about some renal function tests. The first ones I have listed here are tubular reabsorption, or also known as concentration tests. These are all used to detect early renal disease. Osmolality is one, and this is measuring the amount of solute dissolved in solution. I'm used to measuring this in the clinical chemistry department using a freezing point depression system. And specific gravity can also be used for this, and that's when the density of solute dissolved in solution is measured. We can do this using a refractometer or the dipstick pad on the dipstick, and also a urinometer can be used for this as well. Both of these are used to evaluate renal concentrating ability, monitor disease states, and monitor fluid and electrolyte balance. Secretion tests. There is one that is used. It's called the PAH test, where P-aminohippuric acid is infused directly into the patient. And if there is a renal problem, that PAH is not completely removed. Normally it should be, so that indicates that there's something wrong with the kidney system or the renal system. Glomerular tests. This is assessing renal waste removal and solute reabsorbing abilities. If this is decreased, it does indicate abnormal renal function. Probably very familiar with the creatinine clearance test, which assesses glomerular filtration rate. This is a good test because creatinine is not absorbed by the renal tubules. There's also something called the estimated glomerular filtration rate, and this is something fairly new that only uses blood creatinine levels and something called the MDRD, which stands for modification of diet in renal disease. So you do need to correct for gender and race for this test, and results are only reported as a number if less than 60 milliliters per minute are found. This is more of an automation, a test that a lot of the newer automated instruments will perform and calculate. I'm not that familiar with this. My time in the laboratory is mostly entering in the parameters for the patient's height and weight and for the machine to actually calculate a creatinine clearance for me. But I have seen this on many newer laboratory reports. The problem is if your machine goes down, you still need to be able to calculate this out. So that's what we're going to do next, is practice doing a creatinine clearance. I have the formula listed there for you. U stands for urine creatinine value, V for volume, P for plasma. The 1.73 is a constant which represents the average body surface area of an average adult. And SA or BSA, sometimes it says BSA, is a patient's body surface area. Now, in order for us to do this, the patient's body surface area is either going to have to be given to you in the problem, or you need to determine it using a nomogram. So I would say on your boards, more than likely, they're going to give you the patient's body surface area. The other thing that we need to keep in mind is that most of the time, a creatinine clearance is ordered along with a 24-hour urine collection. The desirable collection for a creatinine clearance is 24 hours. But just know that this formula can be utilized for samples other than 24 hours. You can have an 8-hour urine collection, or you can have a 12-hour urine collection, and you can still calculate a creatinine clearance. You just have to make sure that you're taking that number into account. But the optimal and preferred urine collection for a creatinine clearance is 24 hours. So at this time, I would like to go to the e-board, and we'll just practice doing a simple creatinine clearance. I can almost guarantee that most of you will probably be asked to do this on your boards, so it is something that you should definitely be familiar with. And I would suggest simply memorizing all of the formulas, all of the mathematical formulas you could possibly fit into your brain prior to your boards. That's what I did prior to taking my boards. And when I arrived and got all settled and they gave me my one piece of scratch paper, I simply wrote down every single formula I could possibly remember on that piece of scratch paper, and I had them there for when I took my exam. So I'm going to go to the e-board now, and we'll practice doing a creatinine clearance. Okay, so we need to calculate the creatinine clearance given these numbers. The patient is an average size adult. The creatinine clearance is that urine creatinine value divided by the plasma value, multiply that by the urine volume per minute, multiply that by 1.73, which is a constant, and that is an average adult. And then the patient is an average size adult, so we are to assume that their body surface area is 1.73. So if you take a look at this, this is going to end up equaling 1, and anything multiplied times 1 is itself, so we really don't need to do this in this equation. So I'm just going to simply plug in numbers now. The urine creatinine is 120 divided by 1.2 times 1520. Now we're going to divide this, it's per 24 hours, but we want to convert it to minutes because that is the units for a creatinine clearance. How much was cleared by the kidneys per minute? So if you take 24 hours, how many minutes in 24 hours? 60 minutes in an hour times 24 is 1440. Or if you had an 8-hour collection, it would be how many minutes are there in 8 hours? And we aren't going to do this last part because it's going to equal 1. 120 divided by 1.2 is 100. $15.20 divided by 1440 is 1.0555, so I'm going to round up to 1.1. Multiply that out and we get 105.555. I'm going to call it 106 milliliters per minute. So this patient's kidneys are filtering out 106 milliliters per minute. Pretty simple if you break it down and you just remember what the formula is and which numbers go where. Practice these, memorize the formula, and I look forward to hearing from you all at the live Q&A. Tune in next for part 2.